Hello class, we're going to begin some lessons on neuroscience. Actually, the topic is going to be more general. We're going to talk about environmental responsiveness in neuroscience. That is, how living systems respond to their environment. In multicellular organisms, this involves an electrical communication, an electrical type of communication between cells. In advanced organisms, such as us, we have a central nervous system and a brain, and we'll be talking about the basics of how all of these works. This is a very large topic, and so we're just going to be hitting the tops of the trees in our treatment. Living systems sense and respond to their environment. Now, you might ask the question, how many senses do we humans have? You might say 5. But actually, we probably have more. We have more senses than that. It depends on how you count. I mean, you have the fundamental 5 senses of hearing, taste, vision, touch, smell. Forgot smell. But actually, our skin has a number of different type of senses. We can sense heat and cold. We sense it in a different way. Pressure, pain, itch. We also have some internal sensors, which usually aren't counted in those 5 basic senses. We know when we have to go to the bathroom. We know when we're hungry, for example. And even in vision, we have really 2 different types of vision. One involving rods senses the intensity of light, and another involving cone cells senses color. So you could say we have as many as 20 different senses if you wanted to, but oftentimes we sort of reduce those down to maybe 5 fundamental senses. In this lesson, we're going to talk about how living systems sense their environment, and we'll talk about how cells communicate signals between each other. At one point, we'll talk about single-cell organisms and how they sense nutrients, and this is what is called chemotaxis. For multicellular organisms, like our cells, the communication between cells can involve a chemical signaling, such as hormones being passed from one organ to another, or some chemicals pass between cells and their neighboring cells. But the type of communication that we will talk mostly about is electrical communication between cells involving nerve cells or neurons. So a neuron is a name for a nerve cell, a type of cell that can transmit electrical signals. The bottom right shows a drawing of a typical nerve cell. It's kind of a scraggly looking cell. It has a cell body, what is called a soma, the main part of the cell, which has the typical things of mitochondria and ribosomes and a nucleus. But also you see these bushy kind of things on both ends. On one end you have dendrites, which receive signals from other cells. On the other end you have what is called the axon termini, which is the point of transmission of a signal to the next cell. And in between you have this long thin part called the axon, which is analogous to a wire. And in some nerve cells, this axon can be as much as 1 meter long in humans and even longer in some animals. Let's go back for a minute and talk about how this was all discovered from a historical sense. And let's talk about Luigi Galvani. Luigi Galvani was a physician in Italy, and he studied the electrical stimulation of frog legs. He found that a frog leg, if stimulated with static electricity, would actually twitch after the frog was seemingly dead. So he gave rise to this notion that there's some kind of electrical conductivity within living systems. Now we'll see that nerve conduction is not exactly the same as the conduction of electricity through a wire of copper, for example, but it's similar. Instead, nerve conduction is via the movement of sodium ions and potassium ions across a membrane, across the membrane of these nerve cells. A number of the advances in our understanding of nerve cells has been done through studies with the squid axon. The squid axon happens to be fairly large, especially in diameter. It's about a millimeter in diameter. A millimeter is about the diameter of a dime. And that may seem small, but in terms of cells, that's actually a fairly large cell in terms of width. And of course, axons can also be very long. But the squid axon is large enough to be manipulated in a laboratory, and that enabled scientists to do a lot of studies to understand how axons work. I have on this slide a link to a YouTube video in case you're interested. I won't show that because some people might not like to see a squid being operated upon and having an axon removed. But if you're interested and you download the PowerPoint, you could actually click on that. This slide shows how an axon can be connected to two other nerve cells and how a signal can then be transmitted from one to another. The connection between nerve cells occurs at what we call a synapse. That's a junction between two nerve cells. And at this synapse, there is a chemical signal that is transmitted from the end of one axon, the axon terminus of one, to the dendrites of the second nerve cell. And then the electrical signal is transmitted along that long axon to the end and then to another cell or to a muscle cell, for example. This is basically how signals are transmitted from one cell to another, but what happens at these synapses? Let's take a closer look. This is a blow-up of the synaptic junction. If we look at the left first, we see the terminus of one axon. That's the one that's more rounded. And the dendrite of a second axon. At this top axon, the axon terminus, there are what are called synaptic vesicles that contain neurotransmitter molecules. And when a nerve signal is transmitted to the end of one nerve cell, there will be a release of these neurotransmitters. Those are the little triangle-looking shapes. A release of the neurotransmitters, which then cross the synaptic cleft and bind to a receptor protein on the other nerve cell. And when that happens, then it triggers the transmission of the nerve impulse to the second cell. Now, after the neurotransmitters are released and some of them bind to the receptor on the second cell, it's also important that those neurotransmitters are processed and moved back into the first cell, or some of those neurotransmitters are actually cleaved or destroyed so that they will not continue to activate the second cell. That is, you want to have the nerve transmission to be single events, but then to be able to refire that junction and have a second nerve impulse be transmitted. As we said earlier, nerve conduction is an electrical type of conduction, but it's not exactly the same as the transmission of electricity along a metal wire. Instead, neuron conduction is via a wave, what is called a potential wave. Now, what this means is that in a nerve cell, in the middle, I've drawn a long, thin nerve cell. And if you look very closely, you can see on the inside of this nerve cell, you will see a lot of minus signs. And on the outside of the two sides, both the top and the bottom, of course, it's a cylinder actually, you see a lot of positive signs. What this graphically represents is that the inside of nerve cells is negatively charged with respect to the outside. Now, this is generally true of all cells, but it's important for nerve cells. The inside is more negatively charged. In the top part of this graph, it shows something called a membrane potential. And it shows that ordinarily, in what is called a resting potential, there is a negative voltage. Now, don't worry about the numbers, just focus on the sign. That is, a negative voltage means that there's an excess of negative charges on the inside of the nerve cell as compared to the outside. But the point is that it's negative on the inside. The sodium cations are in excess on the outside of a nerve cell. The sodium cations would like to diffuse to the inside because the inside is more negatively charged, but the cell membrane limits the diffusion of sodium cation, maintaining this charge imbalance. Well, what nerve conduction is a transient depolarization, that is, an allowing of the sodium cations to go into the membrane transiently. That then disrupts the membrane integrity and allows for sodium cations to diffuse in adjacent regions, and it essentially propagates down as a wave, propagates down the nerve cell. As soon as the sodium cations are allowed to diffuse into the neuron cell at one location, they begin to be immediately pumped back out. Now, this is illustrated in this drawing by the sort of yellow or gold proteins, these little clumps, which represent what is called a sodium channel. So when a membrane is depolarized, the sodium cations diffuse through the sodium channel to go inside where they want to go, and then they're immediately pumped back out. It may be kind of frustrating if you're a sodium cation because you want to go in, but you're pumped back out. But again, that acts as a traveling wave or a traveling disturbance that moves from one end to the other. Now, this may seem like a very complicated way to transmit an electrical signal, but the truth is we don't have wires. We don't have electrical wires inside of our body. So this is the biological way of conducting electricity. But it can be very rapid. Neuron conduction can occur at a rate as fast as 100 meters per second. Think of 100 meters as being essentially the length of a football field. That an electrical signal can be transmitted that far in a second. So neurons are these biological wires that connect different cells in our bodies. Now, importantly, they will be the connection between sensor cells, and the sensor cells, we will talk about a few, but they could be cells that sense chemicals, they could be cells that sense pressure or photons or sound, and these sensors are actually specialized endings of nerve cells. So the nerve conduction goes from the sensor cell connecting to other nerve cells and eventually to the central nervous system in some cases. This would be responding to an external stimuli. But also nerve cells are involved in conducting signals the other direction. If I want to, for example, click on my mouse, my brain has to send a signal through my nervous system to activate muscles in my hand and fingers so that I click the mouse. So this also involves conduction of a signal through our nervous system. In the next talk, we'll pick an example of one of the senses, actually the sense of smell, and talk about how it works. But first, we'll talk about an even more elementary example of how a single-celled organism can sense chemicals in its environment and respond to those chemicals to actually swim towards those chemicals, a process that we call chemotaxis. And we'll see that that's a very elementary or rudimentary type of sensing of the environment, which we'll talk about first before we then describe one of our senses, the sense of smell. So we'll pause now, take a break, and then we'll pick up with a lesson in which we continue to talk about sensing the environment and sensing chemicals in our environment.